This is Josh Wagner with Montana Department of Ag, the Early Detection Rapid Response Coordinator. And as I was just saying, we can't publish these videos to YouTube and share them for educational purposes if we show the attendees without attendee permission. That's why we have this lockdown. So the Q&A, if you have questions for me or Mallory or Dr. Ransom, just put them in the Q&A. And then in the chat, twice during the presentation, I'll ask for your license number and your name, and you can put that in the chat, and you'll get 1 credit. And then I think it goes across a whole bunch of categories. I'm going to go ahead and apologize for my voice. I got a bad cold this weekend. I might not be your typical jovial Josh, but I still get excited about weeds when I have a cold. And it's 9:01, so I'm just going to go ahead and kick it off so we have more time for Corey and Mallory. So let's try the share thing. Everybody in Montana knows this is my worst part. Okay, so we're going to do a quick Montana update for about 15 minutes, and then we're going to pass it on to these Utah State University experts. But for Montana, big news, Palmer amaranth and water hemp. Last few years, it's been highly on our mind. These are crop weeds, so if you're a county coordinator or someone like me, we work more in range and roadside, wildlands. These aren't necessarily our main wheelhouse, but they're going to have to become that. So you can see prior to 2025, we had Palmer came into Montana in 2023, and water hemp came in 2020. The Roosevelt County water hemp from 2020, it's still hanging around a little bit, but we're keeping an eye on it and managing it. It's a weird situation with a slough and some BIA and some private land and railroad. So we've had a hard time knocking it out, but we very much have it under control and are aware of it. All the other ones we've knocked out. You can see I've drawn a line through those. Both those species are dioecious. So in a lot of those instances, we only had 1 plant, but some of those instances we did have multiple plants and we were able to rogue them out, monitor, and we're doing well. So that all changed this last summer. We'd never really had a whole bunch of sightings. This last year we had lots. Fortunately for Palmer, nothing reproduced. Water hemp, we did find a few acre site south of Glasgow. That's going to be a huge priority. That's probably never going to be able to be eradicated. North Dakota has found when they get over 100 plants of water hemp, they can't eradicate it. So we've got a few hundred acres. Probably not going to be able to get that one. And then since Palmer amaranth is native to the southwestern U.S., and water hemp is a central U.S. plant, we've always been concerned about Palmer, but North Dakota's experienced that maybe water hemp's going to be a bigger threat to us northern counties. It seems to evolve faster to our conditions and evolve resistance quicker and establish faster than Palmer. With Palmer, you'll notice we had multiple bird feeder sites. That's probably the millet and the bird seed. Millet is a big issue, as are custom combiners. You can see we had that too. And then this is a plant here. Let me get my pointer going. This right here, oh, it's not a good pointer. No, maybe that's not either. We'll go with laser pointer. This plant right here was in Miles City underneath the bird feeder. You can see when it's in that smaller stage, this is bigger than a rosette, but when it's small, it's going to be very symmetrical and look like a poinsettia compared to other pigweeds. Here's an example of that. Palmer amaranth, Palmer amaranth, Palmer amaranth, poinsettia. So you see this long petiole, that's another indicator, and then the symmetrical look. So look for that. Long petiole. And then this spring in February, I was presenting in Wolf Point, and a young farmer came up afterwards and showed me these pictures on his phone. And you can see all here on the field edges. Looks like Palmer amaranth. Big long seed heads. This would be a male. So it's dioecious, so the females have a real prickly seed head and the males are really soft. This would be a male right here. We haven't confirmed this in the field yet. We would like to do that in May. But I'd say there's unless he's pulling a world-class hoax, it looks like it's there. It looks like it's been reproducing, you can see, since 2018, so 8 years. So I'm glad I gave that presentation, and I'm glad he came up and talked to me afterwards. Hopefully there's not more hiding out there in Montana, but it's a big, sparsely populated state, so there very well could be. Again, mentioned they're dioecious, so every other pigweed in Montana is going to have the male and female plant parts on one plant. Water hemp and Palmer amaranth are going to have two separate plants. So if you ever see that, we're in trouble. And there's that male, that soft male, and that prickly female. Then, in addition to the long petiole and the symmetrical look of, look for that long seed head. You can see how long these are compared to the short, squatty, smooth, or red root. We do have all of these in Montana. Powell could be easily mistaken. It has the long, slender seed heads too. But we're happy to look. Even if it is Powell, we really don't care. We want to find out. Phragmites, we only found one additional site this last summer. Had to check several sites, but we only had one. So now we have basically two small sites in Gallatin County near Bozeman. And then we have the stuff on the High Line up near Havre. So that's all we have in the entire state. This, you can see there's a native one right here, and you can see that red stem. That red stem's a dead giveaway. You can see the different color too. This is kind of bluish green. This is green, straw colored, seed head, coppery colored. Utah knows a lot about this. If Mallory or Corey want to mention anything about the Great Salt Lake, fortunately, we don't know a lot about it yet, but we have learned how to identify it pretty easily. This is up by Glasgow too. Don't be tricked by how big and tall it can be. Everybody knows how tall Levi is. That's stuff is way bigger than him, but that is native, and even through the winter, it did carry that red color on the stems. Rush skeleton weed. We got one additional grant this year, Beaverhead County, because we found at least 5 new sites in Beaverhead County with just a few plants. And we have these existing grants working on it. And Missoula County, who's covering mineral and granite counties, they're finally going to get working dogs working on rush skeleton weed, which everyone's been demanding for a while. It's great news. They're a huge part of our success with dyer's woad. And a huge part of the difficulty of rush skeleton weed is the difficulty of finding it. So they should really help. As for what we had last year, Beaverhead County is kind of a new scary place down here. It's the biggest county in Montana, and a lot of the sites were high elevation, 7,000 to 8,000 feet. Missoula County is starting to spread from there. The interstate vector right there from Idaho has been a big deal, where it's starting to spread. New site east of Helena, and new sites in Broadwater County. So we've really been trying to keep it to the east side of the continent, or west side of the Continental Divide. It's to the east side now. Kind of scared we're hitting that tipping point, but I think we're still doing pretty all right. Garlic mustard is one, not on the state list, but it seems to be popping up, seems to thrive, especially in town, in forested areas, in trees. You can see the plant zones and the elevations. It's got a pretty wide variety of places it's doing well. Keep an eye out for that. I've given some presentations in the last couple of years about what it looks like, what it smells like. We can go over that again anytime. Success stories. Dyer's Wood, we're just continuing to kick butt. Thanks to Amber and some of the partners, Missoula County and the University of Montana and Sweetgrass County. So we had 327 total plants last year, and almost 200 of those were just in one site. So we're really doing well. Other good news for us, bad news for Idaho is last week we went on a tour of Idaho County, Idaho, which is a massive county, but they have all the way from tall mountains to practically desert. But we have counties like that in Montana too, so it's not unusual for us to have all kinds of different plants. But we saw common crepina, Himalayan blackberry, Turkish thistle, clary sage and Mediterranean sage, sweetbriar, multiflora, and dog rose. And you can see everything green back here is that rose. And then everything right here is trials, herbicide trials they're doing. There's also a bunch of common teasel in there, which we don't have too much of in Montana. This area right here, they'd masticated it, but it was starting to grow back, so they were also going to have to do herbicide work on that. And as we know from like salt cedar work or Russian olive, when you start getting into woody species, and that gets really expensive and really difficult. Then maybe one of the most sickening parts was how much Medusa head they had. In this picture, everything light green here is Medusa head. So from the base of the photo up into here, that's all Medusa head. Then you get some of this darker green, that's something else. But they have a ton of Medusa head in Vent Nada. Something we are also doing well on. We only have 2 sites of Medusa head in Montana. Trying to keep it that way. Yellow star thistle, lots and lots of rush skeleton weed. They almost seem like they almost didn't even pay attention to it. And this is that county right here. You can see it goes from zone 3B down to like zone 7 over here. So there is a wide amount of diversity there of moisture and temperature, but we have the same thing. Like Sanders County right here, we have rainforest. On one part of Sanders County, we have high mountains. Over here, we have single digits by hot springs of precipitation. So we're doing really good in Montana, and we should be proud of ourselves. And if you're on this webinar, you'll probably take part of that, and appreciate you. Vent Nada, we only added 2 counties last year, Judith Basin and Broadwater. Keep looking for it in north central and northeastern Montana. It's a good chance it could be there, but we're not going to jump the gun and say we have it until we find it. We're doing well. I was going to be taking part in this, but since I came down with this fun cold this weekend, Shannon Clark is going to be out throughout northwest Montana doing research sites on rush skeleton weed and blue weed in the coming 3 days, trying to figure out what surfactants and what herbicides work better. We know that sulfonylureas work on blue weed, but we've had a hard time getting rid of it. We know that group 4s work on rush skeleton weed, but we have a heck of a time. We can kill it, but we can't keep it gone. So that's something that, this is why we had Dr. Ransom and Maloree on, because they have more experience with this. We've got these new dash stickers out. If you want to grab those from us and give them out to your people working on the roadsides. Kellyanne has all the different door hangers now to give to your landowners. Reach out if you want those. Woody invasive species management plan has been finished. It's really fantastic, very in-depth. It's real science. Big thanks to ISAN for that. That's been a lot of partners, but ISAN has headed that up. We've got a trifold for education that you can give to your county landowners. And then we've got IPM guides. So we've got all the way from very simple stuff for new people to weeds, new on the woodies, and then we've got the hardcore management plan for us people that are really nerd out on it. And as always, reach out to me anytime, text me, call me, email me, look at my website and familiarize yourself with these weeds that we're watching out for. And with that, I'm going to pass it on to Maloree. So I met Maloree. Well, I met Dr. Ransom with Utah State University, and well, I knew about him because I'm a weed nerd and he's a big name in the weed science world. And he just has a great personality, like so many of the people we get to work with. Really appreciate him. And I met him at the Western Short Course in Loveland, Colorado, and really loved his presentation and just admire him and stuff. So then I went to Western Society of Weed Science in Seattle last year, a little bit over a year ago, and he was there, and one of his students, Maloree McDonald, was there, and she was presenting on dyer's woad and using indaziflam. So I saw a ton of presentations over the course of those days, lots of good stuff, but that was the one that really reached out to me and grabbed me. So I was able to talk to Maloree afterwards and ask her some questions and stuff. And then I told her that we use working dogs on it. So she reached back out to me a few months ago, and we've been trying to brainstorm how we could work together. And then through that, I was luckily able to get the two of them to agree to present here. And so that's kind of the story behind it. And thanks a whole bunch to them. If you will type your license number and your name into the chat, we will let those two take over. Thank you.
Hey, Josh, can I just say I really appreciate the opportunity for us to join you today. And man, I learned a lot just in this 15 minutes. And I've already figured out some things that we can do better here in Utah as far as educational materials and outreach. And we have a lot more in common than we might think. So I think this is just really helpful for us to cross borders and to kind of see the perspectives of adjacent states and what they're doing and where our successes and failures are. So thank you so much for having us. I don't know if you want us to introduce ourselves a little bit or.
Yeah, I didn't ask you for a formal bio. I
Yeah.
know quite a bit about you guys from just knowing you and from seeing your work, but please go ahead and share more.
Yeah, I'll just quickly, I'm the extension weed specialist here at Utah State University. I've been here about 20 years. But prior to that, I spent 9 years at Oregon State University at a small branch station working in specialty crops. And so my experience goes all the way across from kind of small scale research plots and specialty crops to setting priorities for the Fish and Wildlife Service at refuges across the country. And so it's been a really fun 30 years as a weed scientist. But I did my master's degree here at Utah State. And so for me, that's why I left Oregon State, was to come back to what I consider kind of home here in Logan. And it's been amazing 20 years. Maloree started working for me as a freshman here at Utah State and has been in my program for over 5 years. And I'm happy that she just recently accepted to begin her master's program with Morella Ortiz, another weed scientist here at Utah State. So she can't leave us yet. And she's been exceptional and just brings a lot of passion to our program. So I'm going to let her kind of take a lead today. And then I'm hopefully here for backup and to tell a few little stories as we go through our presentation. And hopefully there's some things you can glean from our experience. I'm not sure that we know more than you do about these weeds. We've definitely had them longer, but still challenging. And you're winning a lot more battles, it feels like, than maybe we are. So it's really fun to see that. I guess if I could give encouragement, man, I talked to a commercial operator, And last year, he mashed down 8,000 acres of frag on the north end of Great Salt Lake. That's just one project. So, man, the little you have, it's worth every effort to make sure that that's all you get. Because once it gets as bad as ours, just millions and millions of dollars going into management of that species. So again, really impressive to see what you guys are doing, especially on that early detection, rapid response. But Mallory, I'll let you go ahead, and you're welcome to.
Okay.
Mallory, I think, is originally a Cache Valley gal from here in the valley, but then kind of grew up in Colorado, and then we got her back for education. So we're making her native again here.
That's right. Yeah. Okay. Sounds good. So, um, yeah, like Corey mentioned, I am starting a master's here at Utah State, and my main project is actually with aquatic invasive species, but I just graduated with my undergrad like last week, which is exciting. Um, and during my undergrad, most of my work was with terrestrial species, and namely dyer's woad, and most of that work, all of the dyer's woad work was mentored by Dr. Ransom. So, yeah, I was joking with my husband last night that I'm not brand new to weed science. Like Corey said, I've been working for him for 5 years, and that was my introduction to weed science. But I wouldn't be surprised if many of you have been controlling these weeds for longer than I've even known that weed science was an academic discipline. So, yeah, I kind of like Corey said, I think we have a lot of fun things to share, and hopefully you can learn something. But thanks for giving us your time. So I'm gonna see. It did work earlier to share my screen, so hopefully. Okay. Are you seeing it?
Yep.
Awesome. Okay, so most of the work that I'm gonna present today is about dyer's woad, just because that's a lot of what I have been involved in the past. There's going to be some projects we're working on upcoming that are going to be focused on rush skeleton weed, but Corey has some things he'll share when we get to the rush skeleton weed part. So let's see. Oh, yes, and there's my name. This is one of my favorite pictures of Dyer's Woad, not because it's an incredible stand, but just because it really illustrates how it can ruin an otherwise awesome picture. This was an amazing sunset that I wanted to capture in Cache Valley, and I just can't even think of like the beautiful mountains or the green grass or anything because my eyes just always go right to that horrible weed. Um, a lot of you probably know how to identify it and kind of what its biology and life cycle is like. It typically lives as a biennial, but it can be an annual or short-lived perennial. I have some pictures of the leaves on some later slides, but has elongated leaves and light or white midveins, and then the four-petaled flowers that are typical of plants in the mustard family. And we always say that it occupies disturbed sites, but in our research, we have started to kind of study how it invades dense perennial grass stands. And as you can kind of see I don't know if you can see my cursor, but it is a canopy dominant species. So especially right now, early spring, it's really easy to identify. Um, Corey is really good with his lookalike, so I'll let him add anything he wants to as we go through some of these. But this is a picture of Dyer's Woad on the left, and then fernleaf biscuit root on in the middle and on the right hand side. And the similarities between these two species are mostly in the flower. So if you can see, they both have those tiny white clumps that kind of umbel shape in the flower. If you look closer at the plant, at the leaf, you can see that this biscuit root has a leaf that's pretty typical of a member of the parsley family, which it is. So not a lookalike in every respect, but it is important to note that not everything with tiny yellow umbelliferous flowers is dyer's woad, and some of these are native species that we would do well to protect. So we also have the native water ragwort, and this is more of a lookalike in the stems and leaves. And again, this left hand and center picture is the ragwort, and then on the side over here on the right is dyer's woad. And you can kind of see the lighter mid veins on the ragwort leaf, and also like the pale, maybe slightly purplish stem on the ragwort. And this stem over here on the dyer's woad is dark purple, and I would say that's the darkest purple I've really ever seen a dyer's woad stem. It is also pretty typical for the stems to look similar, more similar in color to the ragwort here. So just another native species to keep in mind when identifying dyer's woad.
Mallory, just maybe one point on that is that ragwort we typically see in really wet areas, and so that that helps a lot too. With we don't typically see the dyer's woad down in those riparian kind of areas, but it has been one that's been submitted where people are a little confused by it.
Yeah. Yeah, thank you. Yeah, as far as movement goes, I think these pictures are really interesting and really telling. Like I mentioned earlier, disturbed areas, we see it a lot along trails and ditch banks. Pictured here is a gravel pit, and you can see it just occupies this whole side of the gravel pit where they've been digging. And then on the side here, this was a swath of land that was disturbed with the placement of a pipeline. So you can see that it just immediately invades a lot of disturbed places. One of our, and by our, I mean Dr. Ransom's working theories, is that one of the ways that it's able to travel is through movement by people and animals. And this is a picture I took just a couple days ago of the seed pod. You can see this is the seed, and there's this outside pod and then just one seed per pod. But the peduncle where it attaches to the rest of the plant is slightly hooked. And so one of the theories is that maybe this can hitch a ride on fur or feathers of animals and facilitate kind of the spread of the species. Yeah, these these next few images are hard to look at. I wanted to include a few of these pictures of some crazy infestations, not only to commend the efforts that are going on to control dyer's woad in Montana, because I've never been to Montana, but just through my conversations with Josh and some of his partners, I don't think you guys have anything like this. But I also do want to reinforce this idea that weeds can take off at any time, and eradication and prevention are kind of ongoing efforts. And in the case of dyer's woad, I feel that there's that old adage that an ounce of prevention is worth a pound of cure. And I think in the world of dyer's woad, I think an ounce of prevention is worth probably even more than a pound of cure. So this first picture I have is one I took at the very edge of our research labs in Collinston, Utah. And it's not an optical illusion. It really was that dense of a stand that in that year basically occupied that entire region of that foothill. Um, and this was in a rangeland setting. We, like I mentioned before, we're often saying that it occupies disturbed areas, which it does. But it's interesting that this weed was able to overtake what to my knowledge was an otherwise healthy perennial grass stand. So I'm not sure what the management was like before we did our research here, or if there was a lot of disturbance, if they did certain practices that would have introduced so much seed, or yeah, like I said, not sure what the management was like, but it is interesting that this otherwise healthy stand was invaded like this. And I will say all the pictures that I'm going to show, this one is in rangeland, and I think the other two are just in disturbed areas. But we also do see it in like perennial cultivated fields like alfalfa fields. This was a picture I took by Newton Reservoir here in Utah. This is at the end of the summer when everything has dried in Sonest, and this is when I think the public really starts to notice it because this is really ugly. I don't know if the public always um, associates the beautiful yellow flowers of spring with noxious weeds, but then as soon as it turns to this kind of eyesore, then there's a little bit of a little bit more care and concern that the public has towards this species.
Mallory, can I interject? One
Of
of the
course.
stories from years ago was a newspaper writer in Brigham City writing about the beautiful yellow flowers in the spring. And then a follow-up article later complaining about why nobody was taking care of this nasty weed with the dark seed pods. And without realizing that they were talking about the exact same species. And we do actually have people who kind of come to Cache Valley to look at the yellow flowers in springtime without realizing that's really nasty, noxious weed.
Hmm. Yeah, I don't have this picture on this presentation, but one of the girls in our lab that's doing a PhD right now, she has some pretty humorous engagement photos that the photographer was like, come out here by these pretty yellow flowers. And Josie, knowing that that was a weed, was like, they're not really as beautiful as you might think they are. But anyway, yeah, so a difference in public perception between flowering and seed set. And then this was also just a picture I took a couple days ago. This is Hiram Reservoir. I grew up, kind of like Corey mentioned earlier, I grew up in Hiram, and I haven't always been super interested in weeds, and so I don't know if this population has been here for this long, and I just didn't notice it because I wasn't involved in the weed world. But this year it was especially striking to me how bad this infestation has gotten by the dam here. Um, as far as cultural importance goes, I'll maybe let Corey weigh in on a fun story he has, but the plant was originally brought here to cultivate for indigo blue dye. It was brought to Brigham City, Utah, which is in northern Utah near Utah State University. And not only does it have the properties of being able to dye textiles, but some medicinal properties have been claimed to be associated with the plant as well. So I don't know. Do you want to tell your story, Corey?
Yeah, if you'll notice, one place you can find it currently is in this airborne, which is supposed to be kind of a, preventative. And so it has been marketed and actually believed to have some antiviral properties. So I think that's why it's in this airborne product. But in China, forever, it's been used for its medicinal properties. And they sell a product there called Mei Len Gen. And Ralph Whitesides and I were actually approached by a group from China. They were based in a startup area in Greenwall, Texas, but they came because they saw the Bag of Woad program and thought maybe they could actually take the dyer's woad that we are bagging and use that to create their supplements for sale in China. And they have plenty of it in China, but they were collecting it from really dirty, kind of polluted roadside settings. And so this idea of it coming from a pristine mountain environment, like with the opening slide picture, they thought would be amazing marketing. And Ralph Whitesides and I thought that if indeed we could develop a commercial market, that could be a possibility for incentivizing people to go and remove it. And so we kind of entertained that. And as part of this deal, they asked for us to collect for them 100 pounds of dyer's woad root, 100 pounds of dyer's woad leaves, and then later 100 pounds of dyer's woad seed, which they had the permission of the Texas Department of Agriculture to bring into Texas. And we actually got a letter from the Utah Department of Agriculture saying that it could be sent to them, with them realizing that this is a noxious weed. So probably not the best idea I ever had as a weed scientist. Definitely an ethical dilemma for me. But after we even bought a washing machine so we could wash the roots and the leaves and air dry them so it was, suitable for human consumption. And when we had it all packaged up to go, we had the final conversation I had with this group was, when are you going to send us the product? And I said, as soon as we receive the agreed payment. And radio silence. So for probably 12 or more years, I had these big packages of all this stuff we'd collected and was ready to ship off to them that I couldn't bear to get rid of because I'd spent thousands of dollars in collecting and washing and packaging and never heard from them again. So there's what happens, get that contract up first. But I'm really grateful that we never sent seed out of state. I think that would have really caused me heartache in the long run and probably maybe would have caused problems in Texas if they'd actually introduced it to that state. So anyway, interesting. But they were so big, they were telling us that they had actually, their product was going to be so good that they actually hired the person who created the logo for Ferrari to make them a new logo so that they could, improve their marketing. And they kind of said, well, we'd love to offer you stock in the company. We had to explain to them that university professors don't get to accept stock from private corporations for work. And so, and maybe that was the only money they ever really intended to offer all along. But wouldn't it be wonderful, right? If there was such a market that people would go pull it and, we could get it off the landscape. Mallory has something about the Bag of Woad program later, and I'll comment there. But we do worry a lot of times it's kids going out to the middle of a patch, filling a bag of the tops of dyer's woad plants and getting their money. And you do worry, is that a positive educational experience, or are we actually teaching them that it's impossible to control dyer's woad? So something we struggle with here in Utah.
Yeah.
When Mallory said with dyer's woad, an ounce of prevention might be worth more than a pound of cure, that was when you have to tell that story in the future.
Yeah.
Yes.
I did the
100
math
pounds or
today, and 100 pounds of dyer's woad root, if we were looking at the little tablets on the left, would have made 45,000 tablets. So there was, maybe they were hoping to sell those and then get their $10,000 worth of value and then pay us for what we had hand harvested. I'm not sure, but pretty high value again. But probably wouldn't, you could probably overwhelm the market if we pulled all the dyer's woad in the state and turned it into supplements. So probably not very practical.
Okay, so as far as distribution goes, it's not really touching the southern, well, parts of the southern part of the state, yes, but mostly it's a problem in northern Utah. You can see from these ed maps that there's a lot of positive points, and we are treating and to some extent eradicating, but it's still just everywhere. UDAF has a dashboard, which is shown here on the right, where you can see, I don't know if Montana Department of Ag has any sort of similar feature, but you can see here the different projects going on with the species and in what year they began. So Corey brought this to my attention while we were getting ready for this presentation. I don't know if there's more you want to say about that dashboard.
I would just mention that anybody can access that. And so if you'd like to go in, just search UDAF Noxious Weed Dashboard, and you can look at projects by species, by year, kind of shows the polygons and the treatment areas, and just a really nice way that Utah has been able to show the legislatures where the invasive species mitigation funding goes every year and some of the success stories that they have had. Awesome. Great to see that Montana's dashboard there, and that's something that I'd like to take a look at after.
Okay, this is a lot of work that Corey did mapping and modeling before I started working for him on some of these projects. So on the left here is a map of a project that did mapping for the Forest Service. So in the red, I don't know if you can see the key there, but that's the Dyer's Woad mapped in 2007, mostly along this edge right here, but a little bit over here. And then the blue just shows the Dyer's Woad mapped in 2014, so 7 years later. And you can see that there's additional infestations that are starting to make its way through trails and through canyons. Again, probably moved by people and animals. And this is some work that I think I feel like one of the hallmarks of an awesome career in weed science is that you just have so many projects that we were getting ready for this presentation. And Corey said to me, I forgot that we had done this work with the Forest Service. So here on the right is the map that he showed me. And again, he can amend this as needed, but the Forest Service land is outlined, as you can see with these kind of colored splotches. And inside those outlined areas, they used this model they created to predict the probability of Dyer's woad infestations. So the darker the kind of like rust color in here is the highest probability of a Dyer's woad infestation. And then those purple dots are actual infestations. So, I mean, as far as models go, I don't know if I don't have much experience with models, but I've never met a perfect one yet. So, I think as far as models go, it could use some refining. You can see there's some that have no predicted probability, but they're still infestations. But it's still pretty good as far as we did predict. When I say we, I mean they. I wasn't here for this, but predict some infestations where there actually were very heavy infestations. So I don't know if you want to add to that, Corey, or not, but
No, just the challenge that, the modeling is only as good as the data that goes into it. And that's one of the challenges. When we're using EDMAPS data, we don't really have areas where we know the weed has been, but it doesn't survive. It's more or less where it is and it's thriving. And so I think we often miss out on that kind of negative control for the model of where you would not expect it. We do see Dyer's woad across any elevation and pretty much any habitat type. So I'm not sure that it has many limitations, which makes it pretty hard for a model to be helpful.
And then this is another project that I'll have you comment on. I think work like this is really interesting and important. Corey did this to kind of explore the possibility of mapping on a mountain bike with a GoPro and how that could potentially save time from people mapping on foot with iPads. And I think this work is really important, not only because not only does it save time for the people who are mapping, but I think it's also just a really interesting project that is like really intellectually accessible for people who might not normally be interested in weeds. I think it's really an interesting project to kind of get people aware and interested.
And you can really take this to the extreme. Actually, I found myself watching mountain biking YouTube videos, and there are species which I think if I had the track log, I could map based on just somebody in the public who rode a trail with a GoPro on their helmet. So kind of fun. You have to, as a As a scientist, you have to make sure that you try to meld together the passions that you have. And so um, people say you just wanted to get paid to mountain bike. And yes, I was on the clock for a couple days of mountain biking. But we did find that in some situations, we could save 50 to 70% time using this technique, even with the people in the office watching the videos and developing the maps. The maps were actually quite accurate and would be more accurate with current technologies. And the biggest reason we started this was that when we were doing large scale mapping for the Forest Service, if you map in the spring, you get a really good distribution of dyer's woad, but you miss all of the thistles and the knapweeds. And if you wait until the thistles and knapweeds are highly visible or canopy dominant, as Mallory mentioned earlier, then the dyer's woad's gone to seed and much harder to pick out in the landscape. So the idea was, if you're trying to be really comprehensive, if you could map it twice using this technique versus once with somebody on the ground, you would have better coverage in time with the same amount of resource. So that was kind of the thought process. And no, I never got to buy myself a mountain bike on university funds. I used my own equipment for that.
Just to jump in here, Jed Little with Missoula County. I was talking to him last week about it, and he said that you're the OG of it because he does a lot of mountain bike mapping. But I said, who was first? He said, Corey's the OG.
Sweet. I'm going to put that on my.
That's awesome. Okay, so this is kind of some field data which I presented at that conference where I met Josh Wagner. We did an herbicide trial with dyer's woad, and the first thing, even before the herbicides, even before we explored the herbicide effects, we noticed that the dyer's woad responds to annual moisture trends. Not only is it highly dependent on moisture, but it is highly competitive with native species for moisture. So this graph is just showing accumulated precipitation. The dark line is the 19-year average. The blue line is what we actually got that year. And then the orange line is the dyer's woad cover. So you can see in years that we had less precipitation than normal, our dyer's woad cover was extremely low. And then in years, specifically right here when we got over the average precipitation, the dyer's woad cover just exploded. And then this is where the herbicide data comes in. So this is a homemade graph, so I'm gonna just walk us through this and hopefully it will make sense. But this trial was initiated in 2021, and then we went to the field, and twice every year we would count all of the seedlings and all of the rosettes in each of the treated areas. So along the top here in the dark bar, I have all of the times that we went to take these counts. So there was spring and fall of 2022, spring and fall of 23, and then of 24. And then the colored bars underneath represent every time that that herbicide treatment successfully controlled that dyer's woad. And in this case, it's that they successfully controlled rosettes. So for example, 2,4-D plus indaziflam successfully controlled dyer's woad rosettes in spring of 2022, fall of 2022, and spring of 2023. So just so you guys are all oriented to the graph. And as you can see here in the bottom, we did treat with indaziflam alone, and in this trial, by itself, it never significantly controlled rosettes in 2021, in the 2021 trial, rather. So there's a lot of things we could pick apart and talk about this data, but I think the take-home message here is that we had a year of low moisture, fall of 2023, nothing significantly controlled the dyer's woad rosettes because there was no moisture and there were no dyer's woad plants even in the control. So then we come back in spring of 2024 and we count rosettes, and everything that combined indaziflam with a post-emergent herbicide was still significantly controlling rosettes. So you can see 2,4-D, imazapic, and metsulfuron. So yes, this is plateau and escort with indaziflam are significantly controlling rosettes even after all of this time. And this is the same sort of data except for with the seedlings. And again, there's a lot of things that we could pick apart and talk about different idiosyncrasies that happened with this trial. But the take home here is that combining the two different types of herbicides gives you longer term control, which is really important for land managers to know that when they do these mixes, they're going to get control over a more extended period of time. And this is just a picture from those plots demonstrating that breaking that life cycle by hitting both the mature plants and the seeds in the seed bank is critical for long-term dyer's woad control. This is I'm going to show just really briefly some lab data that I did this project in my senior year. Basically, it was just, well, it was supposed to be simple. We ended up doing a lot of troubleshooting to get the kind of experiment that we wanted, but you just mix this auger powder in water, and then I had 9 different treatments. So one of them was a control with no indaziflam, and then the other 8 were varying concentrations of indaziflam, and then I would soak the dyer's woad seed, sterilize it, and then plant it into these little vials of gelatin that all had different herbicide concentrations. And we did this with both dyer's woad and Medusa head. And here you can see in the red graph is dyer's woad. The colored bar in each of these graphs is the non-treated. And then with increasing darkness, that's increasing herbicide rate. And you can see basically, again, the broad take-home message of these graphs is that dyer's woad is at least as susceptible, if not more, to indaziflam as an invasive annual grass, which is what we, typically use to control invasive annual grasses is indaziflam. So it's really encouraging to know that at least on a seed basis, dyer's woad is very susceptible to indaziflam. And these are just some pictures that I took of this trial. We have the long plant in the non-treated control, and then this wimpy little guy. It was the best of its class, but it is still just sad looking at the highest rate of indaziflam. So as far as solutions, we mentioned the bag of woad program, community involvement and education important, and there's still troubleshooting that we can do with those kind of community programs, but it's good to let the community know that they can join the war on weeds. The post-emergent combined with a pre-emergent soil residual herbicide is shown to be really effective in breaking the life cycle and controlling those big infestations. And then this is just a quick picture I wanted to include of the biocontrol rust fungus. Up here is a non-infected plant, and then this is an infected dyer's woad plant with the rust fungus that's obviously not going to go on to produce any kind of viable seed or anything like that. So I think that wraps up dyer's woad. I don't know if Corey wanted to add anything. I know we're getting a little a little tight on time, but
I think we can just follow up at the end if there's questions on dyer's woad.
okay. Sounds good. Yeah, so I'm pretty new to rush skeleton weed. This is a picture of the plots that we put out. I joined Josh's webinar in December and was really intrigued by how anxious everyone is about rush skeleton weed and really wanted to know how I could get more involved. So this is some of the pictures of the plots that Corey put out, I think 2 years ago. You all probably know how to identify it. mostly by its near leaflessness and the spines on the lower stem. Um, I wanted to put in a couple of lookalikes. The rosette lookalikes, these are all weeds, and so maybe you see a rosette like this and you want to kill it no matter what because it's probably a weed. I'm sure there's native species that have rosettes that resemble this too, so identification is important. But just wanted to highlight a few of the similarities between these lobe-leafed rosette species. There are lookalikes that have been charted in EDMAPs. Um, all of the lookalikes are mostly plants that don't have many leaves because that's what rush skeleton weed is. The thorn skeleton weed is a member of the sunflower family, but it has this unique kind of three-petaled pink flower. And then rushy milkvetch, when it flowers, it really looks nothing like rush skeleton weed. Has this pink pea-shaped flower, but here we understand that before flowering, a lot of these species can look very similar. So, and then this is the current distribution. We have much less of it than we have dyer's woad. And what we do have, a lot of it we are treating. This is that same UDAF noxious weed dashboard where you can go in and see the different projects we have and when they were started.
Mallory, I might just mention on that one that when we first, early on, they, the ranch that would be in that center picture, kind of at the core of that infestation, they had 150 acres that they knew of. The rancher didn't want them treating. And so it took about 3 years to gain access. When they came back, when they finally got access, they figured it was over 1,500 acres. So it expanded about 10 times during that 3 year period, which is just kind of tragedy in trying to battle something at the early stages.
Yeah, so there was similar modeling work that we did with dyer's woad to predict the distribution, and this was a greenhouse trial studying the effect of post-emergent herbicides on seed viability. And then I think this trial pictured here, where you can very obviously see this treatment line, was just exploring different herbicides and timing to improve control. And I think I will. Oh, yes, I forgot about this slide. So this is a
You can
picture.
skip the modeling.
Okay.
It turns out it's hard to model in the Great Salt Lake. The wind blows wherever it wants, so pretty tough to model.
And then I'll let Corey talk about this because I know a lot of you are probably interested in specific herbicide effects. So maybe, Corey, would you want to talk about this just briefly?
Yeah. So what we've found, it's really hard to tell the difference in timings because this trial, the spring application was made, and then the fall was that following fall. And here it looked like the fall applications were better. We've done some trials since then where we put the fall one on first and then the spring, so kind of on the same cohort or within the same environmental conditions. And in that case, the spring timings look better. So I think it's just stage of growth and moisture. I think our take homes is that milestone in the spring looks really good. And then Tordon tends to be more consistent, and they're actually using that more in their fall treatments. And then we were just curious, in the greenhouse trial, we sprayed different plants with different stage of flowering. And we found that all of the treatments of milestone reduced the number of seeds produced, even if it was in full bloom or that it bloomed and the flowers had closed, by about half. But if you applied milestone before the flower had opened, we also saw that the seeds that were produced, only about half of them were viable. And so we're just kind of wondering about these treatments when it's late and it's already in flower. Are they impacting seed viability? And yeah, only slightly, unfortunately.
Perfect. So as I mean, as far as solutions go, I think we all know we're back and forth between the drawing board trying to figure out how to better control this species. Animals will graze it. We're still looking into more herbicide treatment options. And then here on the very edge, the right edge here is, I believe this is a picture of the biocontrol insect, a brush skeleton weed infected by the insect. Is that right, Corey?
A mite, yeah, that's just kind of, as I understand, naturally occurring. And we just see it randomly on plants here and there. So
Mhm.
not sure there's a future for treatment with it, but I believe there's some work maybe going on looking for potential biocontrols, and we're hopeful at this
Yeah.
point.
Yeah. So with that, we will take any questions, but thank you for having us.
So we do have a few questions for you guys. Just if you haven't typed in your license name and license number one final time, please do so now. First question is, are the gravel pit and pipeline managers required to treat dyer's woad infestations?
Yeah, I saw that earlier, and that's a really good question. So it is a listed species, but like in Cache County, I think it's so widespread, it's been hard for the county to say you have to control it when we can't actually get to it on everybody's land. So that becomes an issue of law, right? If you're going to apply a zero tolerance, it has to be everywhere, and it is everywhere. And so we've kind of, at least in this county, lost that battle. Um, they've had some resistance from those in the past, even when they've sent letters. But I know at least Box Elder County this year now they are actually, the county is doing the treatments on the gravel pits. So that's a move in a positive direction. It obviously is a critical point, right? Because everywhere that gravel goes, so do the seeds. Hopefully that answers that question, but it's a tough one when it becomes so widespread that there's not enough resource even within the county to treat it. How do you require private landowners and others to do it? That picture along the railroad, they did a couple of years after that finally pressure the railroad, and they treated the stripe all the way through the middle of the county. But again, it's really kind of out of control here.
Well, thank you. Next question is, what have been some of your favorite findings and approaches in the lab and in the field?
I think we're just really excited about, when we started working with indaziflam for annual grass management, I had that idea of how about breaking the life cycle of other species, by putting that long residual down. And so I think we've been really excited about dyer's woad and the fact that its seeds are very sensitive. And we've actually seen some commercial applications where we've gotten 3 to 4 years of control of dyer's woad on large scale treatments. Whereas before, they were going in and spraying and killing those mature plants or those bolting plants. But then that very next fall, the seedling numbers just rebound and reestablish that population. And so that cycle of having to retreat sites again and again and again, I think just, wears people out. They don't feel like they're making progress. And so we're kind of excited to see as that indaziflam goes out on more and more acres, does that lessen the amount of retreatment and allow them to focus on maybe new infestations. And then Mallory's work just showing in the lab that, yeah, it is very sensitive, the seed. We don't see the long term that we see with those annual grasses, and I'm not sure why. It might be that some of that seed is protected in those little seed pods or something else, but at least there's potential there to really help.
Thank
Yeah,
you.
and I can,
go
sorry.
ahead.
I was just
No.
going to add, we got the field data first, and so then it was really interesting for me to do the lab project and show that dyer's woad is very sensitive to indaziflam. And then you can kind of start asking like different questions about what that means for the field data. Like, so like Corey said, like, then why might not this specific treatment be working if we know that on a seed level, dyer's woad is very sensitive to indaziflam. So I think the lab and field data, it's really rewarding to see how they kind of bolster each other.
I might just mention we've also carried that to some other mustards. So Sahara mustard in the southwest, we're having exceptional control on that annual in that desert landscape. It's phenomenal. And then we're also seeing significant improvements in long-term control when we treat garlic mustard in tree understory. And so again, that's been one that's been really frustrating with retreatment year after year. So again, starting to address the seed bank just kind of changes that dynamic a lot.
Oh, awesome. Great ideas. At least like something for the garlic mustard. That'll be interesting. Next question. For the rust fungus on dyer's woad, how widespread is it, and is it helping manage dyer's woad? Second part to that is, how does it move, or does it typically stay in one location?
Yeah, the rust fungus, we've known about it forever. Even in the 90s, when I was here as a graduate student, another student was working on that on dyer's woad. They found it was pretty impossible to create infections by it. It seems to randomly infect one plant while 4 around it are uninfected. In fact, you'll see a plant that's heavily infected and one stem of that plant will go up and produce seed, flower and produce seed, while the other, 6 stems are highly impacted. So not really something that has much potential. Amber Mendenhall, who goes by the Utah Bug Girl, she's our biocontrol specialist. She's amazing. She has shown that she actually collects plants with spores on them, dries them, grinds them up, and if she sprinkles that heavily onto small dyer's woad rosettes, sprays it with water out of her backpack, and it happens to be in an area with kind of higher humidity, like near a stream or in the shade, she gets about an 80% infection rate. And I always jokingly say, if I'm going to spray a plant with water out of a backpack, it's going to have herbicide in it. I'm not waiting for 80%. I'm killing it right then. But I do love how she points out in really sensitive areas, maybe that could be something a person could use where herbicide's not an option, where it is really close to water. But it is just highly variable, as with a lot of those biocontrols, unfortunately.
Well, thank you. Next question is, have you seen any difference in using chlorosulfuron versus methylsulfuron on dyer's woad?
I believe that we only had one trial quite a ways back. And yes, I think that we see, because chlorosulfuron has a longer residual in the soil, I think we've seen some advantage to that. We definitely see that on other mustards like hoary cress or pepper or white top. We see much better control maybe 3 years out with the treatment of chlorosulfuron compared to metsulfuron. A lot of our managers are really hesitant because chlorosulfuron still costs 2 to 3 times what metsulfuron does. But I think that if you were going to use indaziflam, though, it wouldn't matter because you're relying on the metsulfuron or chlorosulfuron to kill the existing plants, and the indaziflam would be carrying kind of that residual. Although we have not looked at that combination in our more recent research with indaziflam. So maybe there's even a better synergy there. Good question.
And so far, the final question, if the last call for questions, type them in the chat. So last question that we do have currently is it is interesting that applications by Milestone impacted the seed viability. What reduction did you see in seed viability, and has there been any research on other species, such as if Milestone impacts seed viability on spotted knapweed?
Yeah, that's a really good question as well. I think, way back, Matt Ranella in Montana did a lot of that work with Milestone on annual grasses. We also had some field trials where we showed that you could treat Medusa head in full head with the seeds. The seed had emerged with Milestone and basically reduce almost seed viability almost 100% on that annual grass. And so it was really based on that work that we thought, what does it do on dyer's woad? And again, Milestone at any point in that flowering process, whether it was in the bud stage, whether it was open flower, or whether it had flowered and closed, so those seeds are actually starting to develop, we reduced seed numbers by 50% compared to untreated plants. The only ones that reduced the viability of the seeds that did survive was before that flower opened. So we actually staged all of those plants based on what the flowers were at and kind of harvested those individual flowers. So really intensive work in the greenhouse. And even that one, though, only reduced the germination by 50%. So untreated had 88% viability. That specific timing of treatment had 40% viability. So not really practical. I think we all know that if you pull dyer's woad up that's in flower and lay it on the ground, it will utilize the reserves that are in those stems, even though it's not rooted anymore, and produce viable seed. So we know that if it's in full bloom and you pull it, at least some of those seeds will go ahead and form and be viable. And so that's, again, another kind of factor that we're thinking about even if we're late with treatment, is there some effect on that seed? And unfortunately, not like it is with those annual grasses.
Well, no other questions came in, so that's all we have for those, and thank you for answering those.
Thanks for sticking with us. We're just grateful to be able to have the opportunity to share. And
I've got a question.
yeah,
you mentioned kosher trials before we started recording. How bad's your resistance down there? We're getting northeastern Montana. We have almost nothing that can get it post-emergent. So, how
we
are you
used to
looking with kosher?
we used to joke in the class that when you looked at the herbicide resistance reports, the Ian Heap site, that we only had one case of resistance ever documented in Utah, and Idaho had like 14. And we always said, that means that Utah is just better at managing our weeds. But the reality is we didn't ever have anybody doing the basic work. And since Eric Westra and Mirella Ortiz have joined our team, Eric has a big history, and we've been able to feed materials that we'd collected over the years with suspected resistance to him. And he's gone through and screened them. And yes, ALS resistant statewide, high levels. Now we've documented glyphosate resistance, not just in the orchards where we originally collected, but across in various locations across the state, much lower percentage, but it's out there. And yeah, so it's a tough one. We haven't really got into the growth regulators that much, but I think some of Eric's screening is showing, yeah, some pretty widespread group 4 resistance as well. So no, we have areas where we know the roadside managers are having a really tough time. Yeah, I think going to things like diquat and some really harsh things trying to get a handle.
It's
We've pushed a lot of it back to some pre-emergent type treatments, right? Which gives different modes of action and a different stage of treatment.
that's where we're at with basically pre-emergent. And the guys, sometimes you talk to the farmers and they say, well, we know paraquat will do it. And it's kind of like, well, I wouldn't want to be
Yeah,
messing with paraquat.
right. Yeah, no, it's getting very, very challenging for sure.
um
I
yeah,
might mention we found a farm this past year with Palmer amaranth We'd had a
Oh,
few cases of single plants, no seed production, and then we found one that they've had it for at least 5 years in a fully reproducing population. Eric's already screened seed from that population and identified resistance to growth regulators, ALS, some PPO inhibitors. Surprisingly, in the first run, not glyphosate. So I don't know how we would have, inherited resistant plants that weren't glyphosate resistant. We hope that holds to be true. But that was just like 6 total plants. We had such limited seeds, so we'll see how that plays out. He continues to test those, and I think we ran out of seed, so there will be plenty from that farm this year. Still trying to figure out how that management goes with UDAF and the growers. Actually, private industry has stepped up and donated some product already to help manage it on that farm. So there's at least awareness, but the people who have it, yeah, we've had a hard time even getting a meeting with them. So
well, his dad's a legend too, and I'm a Colorado State guy, so really
yeah,
gotta give him a shout out.
I would just say they're doing phenomenal work. Morella Ortiz is in aquatics. Really, that's her specialty, and a basic lab where we're doing translocation, absorption, metabolism work. And then Eric's just he's masterful in the greenhouse and in the screening and in the ag world. So really excited about our team here, along with our exceptional students like Mallory, as you've seen today. So
Yeah, sounds like you got a great team. That was cool, Mallory, that you did the test on the seeds in the lab with indaziflam. That's super interesting and encouraging that it's as active as it is, or more active than an annual grass. So
Mhm.
thanks for what you guys do, and thanks for taking time during the field season to come do this for us.
Of course. Thanks for having us.
Yeah. So
yeah, thank you.
next thing is ask my boss when I can take a trip to Utah. I'm not kidding.
It's really important, right? To see sometimes you forget how important what you do is. And so I hate that we're a poster child, but it's definitely important to see what the problem could become if we lose sight of what we're trying to accomplish. So yeah, we'd love to have any of you come down and show you what you don't want to have happen, and maybe encourage you that what you're doing makes a difference, right? It's really, really important to get a handle on these plants before they get to the levels where you can't, right? Frustrating. We live right here in the epicenter, and there's very little we can do at this point, but hope for, new tools to come along. So
Well, thank you, and we'll really appreciate it, and we'll hopefully be continuing to stay in touch. Thanks for everyone for joining, and thanks for all you do. Let's knock out this dyer's woad together. Thanks.
That's right.
Bye.
thanks. See ya.